collect your data for your project. And some of the things that we know from our research that we did in class and with the scientists were what are some of the things that you know as you're coming to Silverbow Creek here at Rocker?
Well, we know that there's a sewage treatment plant upstream that could be impacting this restored region of Silverbow Creek.
Okay, so go ahead and put that in, what you know. What else do you know? What else did you find out as far as where fish are located in Silverbow Creek? Because one of the questions that this class had is that there actually wasn't any fish in Silverbow Creek, but we discovered something about that assumption, and what was that?
We found out that downstream Silverbow Creek there actually were sculpin in the stream, which are fish.
And then there were also some trout as well, right?
Yeah.
Okay. And I don't recall exactly what the counts were from the biologists, but downstream there's actually a pretty good population of trout, so sensitive fish as well. So not just fish that are tolerant, but also some sensitive fish. Okay. However, we lose those fish as we come further upstream, closer to Butte. So our question is really, what do you want to know about as we're out here today?
Well, I want to know why, if this part of the stream is restored, then why aren't there fish here? Because there's fish downstream where it isn't restored.
Okay. And then what were the 5 C's? Do you recall what the 5 C's were?
Cold, clear, complex, connected, and clean.
Okay. So do you think, which one do you think we might have a problem with upstream up here, based on your previous research?
Maybe cold.
Okay. What else might be a problem? So we think cold?
Complex.
Maybe complex?
Maybe complex because we don't know what the macroinvertebrates are like in this stream.
Okay. And we also don't know what the inside of that stream bed looks like right now either, just looking at it. We know it's been restored, and we know we'd assume that we've done some good work with that, but maybe there's a problem there as well. Okay. What about vegetation or soils or cover around the stream? What does it look like out here as you've made your observations?
Well, as I was walking around, I noticed that where the tailings have been capped, there's kind of a lot popping out in certain places.
Okay.
So that could be a problem too.
Okay. How about vegetation-wise? How does it look like out here so far? Just basic observations.
It looks like there's a lot of grasses growing, and the understory looks pretty good, and it looks like we have willows and trees, but I don't know.
Okay. Well, that's a good start, and I think we need to start answering those questions with some real hard data. So we can make some assumptions, but we need to gather some data. And so Dr. A and Bev helped us set up our experimental design for the project, and we're going to look here at Silverbow Creek on Rocker, and then we're going to go downstream where we find fish at Miles Crossing, and we're going to compare the 2 sites.
Okay.
The specific data that we need to collect is some water quality data. So we're going to look at conductivity, which you and I will define a little bit later. You can let that be just something that you want to know more about. Dissolved oxygen, pH, temperature, turbidity. Then we're also going to look at the metals, because like you pointed out, there might be a metals problem here still, even though we've got restored. And then we're going to look at ammonia, nitrate, and nitrite. All right, do you know what those are?
Don't those come from the sewage treatment plant? Is that the reason that they would be in the stream?
Could be. So we're going to answer that question. So put that on one of the things I'd like to know. We're also going to collect data related to the macroinvertebrates, and this time we're going to do it more quantitatively. So we're going to take 5 samples from 5 different sections. So we'll do each bank, the middle of the stream, and then take 2 more. We'll kick for 5 minutes, so we're using the same collection method each time, and then each sample will be separated into these 5 tubs. Then we'll count for 5 minutes. So whatever we find in 5 minutes is our count for each tub. All right, and then I'll show you what to do with that data next. And the last thing we're going to do is a riparian area vegetation assessment.
Okay, Venice, so now we're going to do the riparian vegetation assessment, and you know, you can do this in different ways. We're going to specifically do the green line.
Okay.
And the green line is
So Ivana, we've looked at the vegetation, we've looked at the macroinvertebrates, we've looked at the water quality. Now we need to look at the actual composition of the stream, and we're addressing that complex issue that we're talking about. Is the stream complex? Are there places for things to live in there? So what we're going to do is stretch our meter tape 25 meters. And we're going to start upstream and work our way downstream. And then we're going to zigzag from bank to bank. And as we zigzag across the bank, from bank to bank, we're going to measure if we have silt, sand, or a pebble.
Mrs. Knoll, how does a pebble count tell us about the complexity of the stream?
Okay. Well, when you're in there, where do you think you would find, you've already gathered macroinvertebrates. Where do you think you find most of those macroinvertebrates?
Under rocks.
Yeah. We find a lot of them under rocks. We do find them up in the top layer of the water and in the middle layer of the water, but rocks provide cover and shelter for macroinvertebrates. If you were a tiny baby fish that just hatched out, where would you hide in the stream?
Near some sort of shelter.
Yeah.
So that it would be protected.
Right, so you don't want to get eaten by a bigger fish that's coming down the stream. So rocks and complexity inside of the stream is also not only for where the macroinvertebrates are hanging out, but where those baby fish are hanging out as well. So that's another piece that we're looking at, because maybe we're missing the complexity here to provide good habitat. Okay?
Hey, Mrs. Knoll, why are the rocks here blue?
Well, this is something we're going to write on our chart, because as we're making our field observation, we notice this site right here that there's a lot of bare ground and we have the blue rock.
Yeah.
So something's up, isn't there?
Yeah.
Like, that's not normally a color we see in nature.
No.
So we need to record this on our map, and we have those Google Earth maps that we printed out at school. So you're going to Ivana, on your KWL chart, we know about the sewage treatment plant, and we know about that there are trout downstream but not here. And you know about conductivity. Some of your wonderings were, could the nitrates and nitrates be coming from the sewage treatment plant or affecting what's going on? Would dissolved oxygen be different at night? So why don't you tell me some more of those wonderings? What are some more things that are coming up for you as you're gathering your data?
Well, like we said down there, I was wondering why the rocks are blue and why there's grass on the ground only in like certain sections, and then there's just these sections of bare ground. Like, what is that?
Okay.
And then I was also wondering if the turbidity would be different like during different seasons, because during the spring you think that it would be better because all the snow melts and carries sediment into the stream, and I know that from some of the things that we did back at school.
Okay, so you think it would be higher in the springtime than it is now?
And then I also know we got a dissolved oxygen reading of 8, but we also know that this part of the stream doesn't freeze in the winter time, so I wonder if the dissolved oxygen level would be lower or higher during the winter.
Okay. And why do you think it doesn't freeze during the winter?
Maybe because of the nitrates and nitrites, or the temperature's too high.
Okay, so the temperature is staying up, obviously, because it's not freezing. So the question is, why is that temperature staying high enough that it doesn't freeze down here? Okay. And then, did you ask Bev about the relationship between dissolved oxygen and temperature?
Yes, I did, and I found out that what I learned was that the lower the temperature is, usually the more dissolved oxygen, the higher level of dissolved oxygen you have, which makes sense.
Okay. Another thing that we talked about in the stream that, you know, I don't see you have written down here, is that with complexity, remember those boulders and the bigger pebbles, as water moves over those boulders and bigger pebbles, it kind of aerates the water, so it allows it to touch air more and get more dissolved oxygen in it. And in our pebble count, we discovered we only had actually 60% silt or sand in our pebble count. So we didn't have very complex stream bed here that would allow for more aeration of oxygen as well. So that could be something that we need to do a little bit more research about too. So we'll think about those questions, and we'll ask the scientists what their opinion is as well.
Okay. So Mrs. Knoll, the first thing that I noticed about this site is that, first of all, there's no overstory at all. I mean, there's grasses on the ground, but there's nothing above our heads. And it looks like there definitely are it looks like there's a better pebble count of bigger rocks, and rather than just sediment bottom like the other site was. But why are the fish here and not over there? Because this one looks like the vegetation over there looked a lot better than it does here.
So Ivana, we're at Miles Crossing, and here, this is where the biologist fish shock data tells us that there is sensitive fish or trout here. We've also heard that there are fish have been caught here, and then further downstream from here they've also been caught. We know this is a restored section, and a recently restored section. So what did you expect to find when you got here?
Well, when I got here, just based on the data that they were catching sensitive fish like trout, I expected the site to look like the site we were at Rocker. Where here, when I was going through my vegetation, I had barely any overstory. And there's I didn't find any pollution tolerant grasses, but I expected it to be more for if there's that sensitive of fish.
Okay, and that overstory helps. And I think you said something at our other site too about sampling, and what did you learn about sampling?
I don't know if one sample is good enough for determining, like, coming to a conclusion about why they're here and not there, because there's a lot of variables included, and there could be a lot. The readings at the other site could be a lot different at night than they are in the day. They could be a lot different during the winter, or the spring, or the summer, or vice versa. And if we don't have quantitative data, which is a lot of samples, then I don't think that we can determine anything.
Okay. And I do think you're right on the right track there for sure. So I think the answer to our questions today is we have to collect more data.
Collect more.
Okay.